Janne Lappalainen. Jan builds simulations of neural circuits with machine learning tools to understand the principles of how circuit structure leads to neural computation in intelligent systems. In particular, his work with researchers from Janne Research Campus focuses on realistic connectome constrained models of the fly visual system that detect motion. Please join us in welcoming Janne Lappalainen.
Hi, thanks everyone. Thanks for introducing me. Great. So I'm a PhD student at the University of Tubingen, and I'm talking about the work that I did together with Srini Taraga and Jakob Macke and a bunch of other colleagues from Janne Research Campus. And yeah, I think Philip already really nicely set the stage. So here, this is a very complementary research where we ask how can we go from connectome to computation with machine learning to predict the neural function. So the motivation is that we wanted to understand detailed neural function, and detailed neural function is largely determined by how neurons connect with each other, basically the wiring diagram, right? And then these wiring diagrams, they constrain how electrical signals can pass through these neurons, and that determines neural function in a detailed level. And that is one of that is at least a very important scaffold of neural function. So as a first step, we believed it's good to understand how we can use this wiring diagram to predict function. And so, for instance, here we have the fruit fly, and I'm going to go to the next slide, where here's one example. So if we look at this very complicated sort of circuit on the left, the question simplifies in a very simple way expressed, basically, is how can we predict these dynamics of this neuron that we see on the right, which depolarizes to an edge that is moving upwards, but it is silent to an edge that is moving downwards. So this is a motion computing neuron. And this computation in the single cell underlies then more complex behavior. So for example, the fly that rotates its body in response to gratings that rotate around it in an arena, or that moves backwards, for example. And the fly brain, that's actually remarkably efficient. So I think we already heard that it only has 140,000 neurons to survive, and it can do very complex coordinative navigation. So here, for example, it sees a looming stimulus, and then it coordinates all these movements from the visual stimulus. It realizes it needs to escape, and then uses its 6 legs, jumps off the platform and its wings to escape in the right direction to basically not be hit by the stimulus. Here we have another little movie that shows 2 male flies that fight over a food source, and this is also a nice example that there's just a lot of tools available for studying flies. So this is why it's such a nice modeling organism, because we have, for example, the genetic tools to manipulate these flies to behave in a more aggressive manner, and which is actually the case here. We can, like these flies, they breed very quickly from egg to adult in 10 days. And we have now the connectome, we have functional measurements of neurons, and so on. So this motivates really using the fly as the platform to understand how we can use brain connectome to build a functional brain model. And we have a lot of connectome today, actually. So the earlier slide, the introductory slides, showed the central brain, the optic lobes. This is now the ventral nerve cord of the fly. So you see here what Philip mentioned, that it's cut. So basically the head is cut off before putting this under the microscope. But this is the first complete measurement of the nervous system of a fruit fly. And then we have C. elegans, even across developmental stages, zebrafish connectome, and then mouse connectome, and also human connectome to some degree. So the main question or challenge that we kind of wanted to address with our study was how can we get from connectome to computation. And so for us, the computation largely starts at the single neuron, and that is something we can model, right? Like we can, for example, use Hodgkin-Huxley equations to model the voltage of a neuron, and that neuron has some biophysical properties like capacitances and conductances and so on. But then we don't actually know these parameters, right? So how to measure the conductance of every single neuron or the capacitances. And this is a challenge. We have now a large circuit, so there are a lot of parameters potentially, right? And so there's one way to maybe do it, which is something more abstract, inference, right? So basically, if we measure some data, maybe we can infer something about something else. And then there's another theoretical challenge about what we can do with a fixed network structure. And the first one is that we know that different parameters can actually produce similar functions. So that means, for example, if we put different parameters in this equation and then simulate it, then we can sometimes get the exact same electrical signal, basically, as we would get for another set of parameters. And this is also true for connected neurons, right? And then we also know that the same network with different parameters can actually approximate different functions. So it's really unclear theoretically if we can find parameters that constrain computation with connectomes. So to what degree is the connectome a good measurement for predicting something about neural computation? And this is where our research at the Fly Visual System Opportunity, I call it now, came in. So here we really had the connectome available, so of the Fruit Fly Visual System. And this system has a very clear behavioral task, which is motion detection. And there are other tasks, but this is really the main task of the visual system. And there were also a lot of neural activity measurements available. So this allowed us to use this connectome and build a model that has a one-to-one mapping. And then with that model, use inference methods, basically. In this case, we use deep learning to train the parameters of the neurons. So to compute the task that we know the system has. And then we can give the system the same input that we would give a fruit fly, or that experimentalists have given a fruit fly and then reported those neural activity measurements. And then we can compare what our model predicts versus what has been observed in the fly. So yeah, we really try to bridge this question if we can use the connectome to predict the computation at the neuron level for a specified task, neural computation on a higher level. And here we also, I really liked Philip's slide, like the things that we don't have, that they are the same, right? Like we don't have modulators, glia cells, gap junctions, and such things. All right, so I'm going to talk about the Fly Visual System a little to introduce this. So we have these compound eyes. They have photoreceptors in each of these ommatidia. They are organized in a hexagon gonal fashion, and there are 8 photoreceptors, and they are called in with these type names. So typically one has like a couple of letters and then a couple of numbers, and that describes the cell type. And then there are several instances of these specific cell types, and they differ in morphology, connectivity. So there have been various ways to cell typing. So most of this here, this is from an old paper, actually from a Golgi staining. This cell typing is based on really careful manual analysis. Nowadays, one would typically cluster the connectivity of these cells. And so here, this is the input layer. So that's the retina, right? So the height, the light that hits the receptors, and that those receptors, they transduct the light into electrical signals, and pass them down through a cascade of neurons that perform computation on it. So we have different neural pools that are these meshes of axons and dendrites where we have synaptic connections, and they're in total they're in total 5 actually. So there are 4 visible here. There's a fifth one, which is for circadian rhythm and only unrelated to motion detection. But so the first stage is basically a stage that that does sign inversion and contrast and brightness extraction, and then it's passing down the signal to the second stage, and then here we have already inputs to motion detecting cells, which are the T4 and T5 cells. So there's T4 cell types that predict motion of edges that are bright light increment edges, and then there are T5 cell types that predict motion or detect motion to light decrement edges, and then there are so of course this doesn't illustrate all the cell types available. This is a subset, but maybe one can see it here indicated already that there are also large cell types that really that have large dendrites that integrate a lot of electrical signals from all these local neurons and send back an aggregate signal to the central brain for making decisions about navigation. All right, so this is the anatomy, and there is the hexagonal arrangement of these ommatidia, and there's a retinotopy, and we make use of both these features to build a model from the connectome, because the first thing is we can represent the model in a hexagonal lattice, and the second is that we can assign a coordinate in the input plane, so basically in this photoreceptor cell plane, to every single cell in the model, because it's retinotopic, right? That means that every downstream neuron has a corresponding receptor neuron. And this is what we call our hexagonal lattice model. So so each individual hexagonal lattice here is one cell, represents a cell type, and then each individual hexagon inside the lattice represents a cell. And in total, we end up with 45,699 cells, 1.5 million connections, and they are both feed-forward and recurrent connections. So in fact, like, we have a really big connectivity matrix that is informed by the connectome measurement. And so this sort of roughly corresponds to the central portion of the right eye of a female fruit fly. And okay, so how do we determine the connectivity? So you can imagine that from the EM reconstruction, we get these little dendrites and the synapse locations, and then we assign basically we tile the space with our hexagonal grid, and map that to the input plane for the retinotopy, and then we can count the number of synapses that fall into each of these tiles, right? So for example, this is the input to a T4D cell. T4D is one of the motion detecting cells, and MI9 is one of its major inhibitory inputs, and the input is distributed a little bit offset to the motion detector, right? So we find on average, if we average it laterally across the connectome measurement, then we have an average synapse count in these tiles, right? And on average, we find more synapses above the T4D cell than beneath it, right? So and this is a convolutional filter because the visual system is very crystalline. So if we move down basically one T4D cell to the bottom, then this connectivity pattern would also largely be preserved. So that has actually been studied and also reported that there's very little variability. So that's a nice feature, because that tells us that these neurons compute like a convolutional neural network, basically. And this is what we make use of. And so in our study, we identified actually from a lot of data that preceded the Flywire connectome and the recent Janne fruit fly visual system connectome. We compiled this from local reconstructions, and with we I mean Fabian Schopp, so who also has this pilot paper on this. But here we could really, from these local reconstructions, we could compile a model that identified the 64 cell types that are major cells and cell types contributing to the motion detecting circuit. And this is the cell type connectivity matrix, where we represent the total synapse count from one cell type onto another cell type. And then the red squares are excitatory connections, and the blue squares are inhibitory connections. So as in Philip's work, we have access to presynaptic neurotransmitter information and postsynaptic receptor type. So actually that is additional. So sometimes we have, it does not always adhere to today's law, because sometimes the postsynaptic receptors, they make something else out of the neurotransmitter, right? So this is cell type on a cell level. This looks much more like this. So this is the model if we only plot the connectivity matrix corresponding to the central 91 columns of in total 721, which roughly corresponds to the number of ommatidia in the fly eye. And then this accounts for the connectivity of 5,700 cells. Okay, so now we have a model. Now we have a structural description of the fly visual system. Now we want to go to a functional description of the cells, right? And this actually starts from the synapse counts that we found in the dendrites, and used to arrange this model, right? So we can, because of our cause tiling, we can make an assumption that basically in these coordinates that corresponds to the synapse count between a presynaptic cell type Tj and a postsynaptic cell type Ti. At these coordinates, we make the assumption that the synapse strength is somewhat correlated to them, right? And we have the sign, so that's just -1 or +1 between those two cell types. And we just don't know what the actual unit is of this weight. And this is where our coefficients come in. So these are free parameters that we have to infer that roughly corresponds to conductances in the Hodgkin-Huxley model that I introduced on the motivation slide. So these are now the unknown parameters that we want to infer. And this is one scaling coefficient that basically says it scales all the inputs from equivalent pairs of pre and postsynaptic cell types by the same amount. So that is sort of translating these convolutional filters that we measured into some functional units for the task that I'm going to present next in a couple of slides. So then our synapses in the model are proportional to the rectified presynaptic voltage, and that this is describing the transmitter release at the synapse. So here we have a nonlinearity, and then our neurons, they are actually point neurons. So we remove the morphology besides the implicit morphology that basically comes in through this connectivity mapping, right? And then we have point neurons and describe their electrical signals with graded potentials, and that is also because actually the small neurons in the fly visual system, they are only computing with graded potentials, and so they're non-spiking, which has some neat properties, or at least is not as prohibitive as spiking neurons for optimization. And in addition to these alphas here, we introduce the time constants of the neurons. They are also shared across in cell instances of the same cell type, and we have resting potential. So where the neuron basically typically is, in which state when there's no input, and that already is a relatively reduced model, which is neat because if it's more complex, it needs more computation, and in particular, it's neat that the connectome really gives us the ability to reduce it to 734 parameters because we have 605 filters, so 605 alphas, and we have 65 cell types. One cell type we split into two cell types in the model. So 65, and that means in total we end up with 65 time constants and 65 resting potentials. And so that adds up to 734 free parameters. And so without that connectivity, if we would naively just take any basically neural network of 45,000 cells, then it would be a connectivity matrix of 2 billion entries. When we assume something reasonable, like we have local convolutions, then it would be around 400,000. And yeah, in comparison, also AlexNet has convolutions, but it has 60 million, and it can just classify 1,000 images, 1,000 classes on ImageNet. All right, so now that we have a structural description with a one-to-one mapping to neurons in the fly visual system, and we have the dynamic description of the electrical signals in this model. The question is, how do we find these parameters, right? So that they produce anything meaningful, the dynamics. And I guess one way is to make this neural model more closer to the biophysics, and then use biophysical parameters from literature and try it out, or we use this more conceptual reduction and then try to learn these parameters. So that's what we did, and we trained the model on the motion detection task because we know that the circuit learns to compute, or the real circuit is computing motion. And so I'm going to skip over this and show this in pictures, what we actually do in the training. So we have a computer vision task that is called Sintel, and that has a bunch of rendered sequences. They show basically naturalistic movie, not necessarily natural for a fruit fly to see, but it is a rendered movie data set that has, for every individual video, it has the motion of every individual pixel in the video, and that is represented by the displacement vectors, basically of the pixel from one frame to the next in the X and Y plane. And as a first step, we render the video to our FlyEye geometry, which is really just a basic box blur, and then picking the ones which correspond to our hexagonal geometry, and that we can add to the photoreceptors in our model, and then we can forward simulate the coupled system of differential equations. All right, I'm gonna quickly check that you're still there, because I can see you, but yeah, I don't know if I'm losing connection.
We can hear you and see you.
So that means now we have basically activation in these photoreceptors, and that activation is passed down through the connection matrix to the next cells. So there are synapses that go all the way, or that go here, but there are maybe also synapses that go all the way a little further, and like through this through forward simulating it, the input is really driving the activity also out to the end of the circuit, and produces some dynamic neural activity. So because we haven't inferred the parameters yet. This only works by basically guessing parameters. So we guessed parameters, random parameters in the beginning, but then we want to learn them. And to learn them, we actually use backpropagation. So we needed to implement this in PyTorch to make it differentiable. So everyone who is familiar with machine learning roughly knows where I'm getting at. So, and I'm going to try to explain it a little better. So, but first let me say that the output or the activity of these cells in the black square, that is passed to a motion decoder, and that is just a spatial CNN that cannot compute any temporal differences, which would be required to detect motion. So this is really just to a relatively small CNN that just sees these activations one frame, one time step at a time, and then tries to predict these motion flow maps that we have the synthetic data of. And so because we have the ground truth, we can now compute an arrow between the ground truth and the prediction based on random parameters, and then we can use the differentiability of the model to update the parameters, right? We minimize the loss function, which is just the mean squared error between these maps. And doing that iteratively, we find parameters of the model that eventually manage to compute motion from the inputs. And that is one example of such a network. So now we see the dynamics, and we see from the video the rendering, and that is added to the photoreceptors. We see how that drives other cells in the circuit, some doing, for example, contrast inversion, and some detecting edges and so on. And then we have the CNN that predicts this sort of synthetic representation of motion, and we can compare it to the real representation, or to the 100% accurate representation of the data set, right? We don't actually know what the real representation is, or to a degree we know what the real representation is, but you know, this is the computer vision representation of it, and then there's the fruit fly neuron representation of it, which basically happens in the T4 and T5 cells. All right. Okay, here's just another example. Yeah, this one turned out a bit smoother. Might be based on integration time step or decoder size. And the thing is that now we have these models that have the real structure, that have neural dynamics, that can compute motion, but we don't know if it actually computes the motion in the same way as a fruit fly. So that's where we do basically what experimentalists do, right? So now we can do neurophysiology in silico to validate our trained models. And because when we first started this, we realized two trained models would sometimes, you know, not yield the same result, because the task is non-convex. The optimization problem is difficult with backprop of RNNs and so on. It's really hard to train. So sometimes looking at just a few neural tunings, we already saw, okay, these models, they don't match. So we realized, okay, we got to quantify this more broadly. So we trained 50 models to explore basically what the solution space has. And then we get, for example, activations like this to an edge, and we can compute statistics over this. And here we are sort of in a comfortable position, because there's really, I think, almost 30 decades of experimental measurements that we could use to validate our model, and integrate sort of into knowledge that we can use to validate the model, but that we don't use to constrain the model explicitly, right? So we have asked what can we do with the connectome and the high-level task, and so this, I think, is a position that we're in for the fly, and that we're maybe not in for other organisms, but here it works well. So let me tell you how it went. So segregation into on and off pathways. We know from the visual system of the fly, but also from other organisms, that it separates sort of equivalent computations of orientation and motion into on and off pathways. So it can see in various light and contrast and brightness conditions. It can detect the edges of objects and so on. And this is a very early feature of the visual system in the fly, and that can be measured, for example, as seen in this study. So they showed these light increment flashes, just really bright circuits in the center of the screen, or dark circuits, and then measured calcium responses of, so for example, here these laminar cell types L1, L2, L3, and L4. And then they all depolarize to these flashes. And then we can also render such flashes and show them to our model. And if we now basically predicted the correct computation and the segregation, then our model would also predict these depolarizations correctly. Okay, so, and this is in fact what we actually see. So here, the flash response index on the y-axis is a summary statistic of the voltage response. It ranges from -1 to 1, and it's basically the peak response of the neuron to an on flash minus the peak response of the neuron to an off flash, and then normalized. So if the peak response, so if it depolarizes to an on flash, then it would be above 0. If it depolarizes to an off flash, then it would be below 0. And here, for example, are the L1, L2, L3, and L4 cells, and they are all predicted to depolarize to an off flash. And then we have from the other studies collected these properties of other cells, so including the motion detecting T5 cells and their inputs. So they are off selective, and then here T4 cells and their inputs and the retina, of course. So they depolarize to on flashes. And okay, what I should mention here, so like I said, we quantified the statistic of 50 models. So every single dot here in the violin represents one model. We actually here cut it to the best 10 models because the optimization is sometimes really hard. So we said, okay, let's just look at the ones which actually can compute the high-level task, right? And throw out the 40 which are sort of mediocre at it or bad at it. And then there is also the blue one, which is the best performing model, and we can see so that has the lowest task error, and we can also see how that performs. And the most accurate prediction is made by the median across the models. And then we have a lot of other cell types from the connectomic data for which cells exist in the model, and then for which we can also make predictions for this specific dynamic computation, right? Like for how it depolarizes, which is a relatively simple function. So what we really wanted to get at is to evaluate how the model computes motion. That is less simple than the previous one, because that requires a combination of delays and spatial asymmetries of inputs. But so how experimentalists study this is they show actually moving stimuli to the fly in an arena, and then measure the depolarization here with calcium also to either on edges or off edges. And if we would do this in our model, those stimuli would look like this. And then if they show them in different, so 12 different directions like we do here, and then show the peak response, so the peak depolarization of these voltage time traces, then plot them in polar coordinates. One sees that these neurons respond highly towards direction, towards a specific direction. So that's the T4 responses, and T4 splits into 4 subtypes, one for each cardinal direction, all detecting on edges, and T5 equivalently, but for off edges. So if our model, after the optimization, can compute the motion like the fly can, then we would basically get out those tuning curves. Okay, so we showed these stimuli to the model and computed these tuning curves in the T4 and T5 cells, and we find that actually they computed in precisely the same way as experimentalists found in the fly. With the connectome model now, we can even analyze the mechanisms more deeply. So here we see one voltage trace of a T4C cell, which is response tuning to up motion. So that's the depolarization you see here, but it's not responding to edges moving downwards. And then for this same response, we can look at the input currents and then analyze how this mechanism works. So for example, here we see that there's some delayed inhibition. So if we compare the MI4 currents for preferred direction motion versus null direction motion, then we see that here is a big difference. And this turns out to be very consistent with theories of how this works and experimental measurements. And yeah, here we see a simplification, or like the main connectivity pattern of the major cells that are involved in the T45 motion detection, to sort of characterize this a bit. But then on a higher level, again, we can aggregate this into direction selectivity index. So that's similar to the flash response index, where we compute basically the sum over these lobes, and that we normalize. So it's some vector, some normalized, which lies then between 0 and 1. And that basically tells us how directed are these lobes, right? And that's the direction selectivity index, and it tells us across the ensemble of models. So we have, again, 10 models here, all of them predicted to be very high for T4 cells, and then also for T5 cells, they are predicted to be significant. And then across cells for which it hasn't been characterized, we can also characterize this in the model, and then make novel hypotheses for maybe cell types that have been overlooked in how they contribute to the motion detection of the fly visual system. Okay, so now we did find, though, a lot of variability that you can, for example, see in these summaries. And we wanted to analyze them more systematically. So how much are the 50 models different? Is the basic question. And to do that, we came up with this procedure. Basically, after training, we just show them the whole data set. So we get very varied responses over a big distribution of possible inputs. And then we take that out of each model of the equivalent cell, and then we cluster it. And since clustering in high dimensions is a bit difficult, we first projected to 2D with UMAP. One could use probably a lot of other methods, and then we can see here already nicely that these basically hypotheses for how the cell computes cluster into 3 distinct versions, and then we can take these labels back and sort these into groups, and then we can analyze those models individually for the mechanisms. So that's what we did. for T4C, and in the long supplementary file also for every other cell or cell type. And so here we first see we now go from the worst cluster that computes the task worse with an average error of 5.357, whatever that means, but it is worse than these two clusters. And then we can look at the T4 tuning, and we see that it's not tuned to motion, right? So the worst cluster on the motion detecting task does not predict that the T4 cell is motion selective. And then we chose the next cluster and see that the T4 cell is predicted to be selective for downwards motion, which is actually incorrect. The specific subtype is predicting or it's detecting upwards motion. And this is what our final cluster and the one which is also performing best on the task, so it's most consistent with our data constraint, predicts right. And there are a lot of models in this. So this is a very nice result that sort of surprised us, but it was also satisfying to see that we can basically characterize the whole solution space, because when we cluster these responses and these solutions, they are actually just a handful of clusters, right? Like instead of hundreds, okay, they couldn't be hundreds because it's just 50 models, but you know where I'm getting at. So we have only 3. And then we can even go further, right? So we can ask what is the mechanism that supports these 2 solutions, or maybe even the third. And these mechanisms are typically at the first stage of analysis are related to the inputs, right? So we have these 5 major inputs: MI1, TM3, MI4, MI9, and CT1. And then we already characterize their flash responses. So first thing to look at, okay, how do they respond to flashes in these models, right? And then we see that in the worst cluster, actually the MI9 cell, which is supposed to be off-selective, that does not respond in an off-selective way, which leads to these untuned models, because this is a very important inhibitory input for the motion detecting mechanism. And then for this cluster, the MI9 cell again does depolarize to the light increment, but also the MI4 cell is switched. So now the MI4 cell is off selective, and this enables the model to tune downwards for the T4C cell. And then in this cluster, both of them are predicted to be correctly. So what's the takeaway of this? I think it's nice to see that the constraints of one cell, which is now the T4C cell, say we had measured this, would translate to constraining 2 more cells, right? Because that sort of tells us that activity measurements of some cells will also constrain other cells in such a circuit, and that we don't have to measure every individual cell to maybe, find a functional description of the computation. And it tells us very nicely where just our model is not constrained enough, right? So we put in a lot of constraints. We put in the connectome, the reduced neuron model, and the task. But even then, we haven't boiled it down yet. So, but we have boiled it down to a really good degree, and we can explain what is still unconstrained. So now we can basically apply the same analysis to one of the neurons that we also predicted to be not motion selective in terms of the flash response index. So now I'm just going to put that all on one slide. I think I'm also slowly running out of time. So which is the TMY3 cell. And so our model says, okay, it's very likely to be somewhat downwards tuning or left tuning, and with some uncertainty, right? But this is some prediction we made, and I found personally particular satisfying, right? This is sort of the goal to make such a discovery, and now it's up to experimentalists to characterize this and figure out if it's as simple or if the picture is actually maybe a little more complicated than this. All right, so quickly, to some degree, we ask what's needed for the approach to work. And with what's needed, I mean specifically from the constraints that we used. So we had constraints of the connectome and of the task, and we characterized it to some degree. So here we see correlation to some known neural function. So the first violin of each group is this flash response index. Then the second is representing the T4 motion tuning curves, and the third is the T5 motion tuning curves. And we see that the model, which basically I talked about most of the time, that has access to cell-type connectivity, has single-cell connectivity, has synapse counts and synapse signs, and it is task optimized. So it has these two sets of parameters that are task optimized, that has the highest correlation to these known measurements. And if we would not use task optimization, we could so maybe randomly sample parameters in some reasonable regime, right, where the neural activity is sort of healthy, but not as expressive, and it cannot compute motion. So if we would train a decoder on these models, for example, it would not be able to compute the motion, but we can still measure, characterize what the neural computation looks like, and then we already get strong agreement with actually known properties. And I think this is sort of consistent with Philip's result, even though we have, not the right units, and then these, randomly sampled parameters always means we at least have to choose the mean and the variance. And so if I was completely naive, I think these would even look worse. But at the same time, it also shows how strong the connectome is as a constraint for these functions. Now, here in these variants, we at least choose always one connectome constraint that we didn't use, that we sort of then task optimize. So for example, here the synapse count, single neural connectivity, and so on. And the picture is pretty consistent that it just doesn't work. So here we have seen these competing hypothesis for the T4 cells tune, right? And then, for example, if we don't use the knowledge of the synapse counts of the single cell connectivity, then the algorithm always chooses the opposite side, and we get negative correlations. And that is something to bear in mind, that there can be those biases, right? And that we really require those measurements, which we typically have when we have EM. And if we don't use any of those, we cannot predict it. Okay, so we built a computational model of the fly vision system from connectome and trained it, the parameters with deep learning to compute motion. And we evaluated those trained models across 26 studies, demonstrating they accurately predict neural activity at single neuron resolution. And we showed how activity measurements can refine hypothesis from model, where basically one T4C measurement would also constrain the inputs as one example. And then we derived also some novel prediction. I think generally, I made this quick brain dump mind map of this whole framework, basically, which combines deep learning, computational neuroscience, probability theory, neuroscience in some ways. So we have this deep mechanistic network in the center, and we want to interpret computation of an organism and predict those computations, and using various measurements. And then there are all these methodological and conceptual portions that play into it. This is not fully baked, but I think it's interesting to zoom out a little bit, and that also means to look at the limitations. So one is for sure how to translate this to other circuits, but also to morphologically detailed neurons. So for example, in the retina, motion detection requires morphological detail, and to spiky neural networks. So because optimization of spiking neural networks is typically harder, but it does exist. But we don't have yet shown that it also works. So that remains to be demonstrated to some degree. So here's research or a tool basically that has been developed by Michael Deisler, colleague, and Jakob Mackes group, and it's called Jaxley, and that is specifically to simulate morphologically detailed spiking neurons and create networks of this that are differentiable and that can be similarly optimized using deep learning. And then here we basically demonstrate that we can build a complex network with complex morphologies. These are just CR1 morphologies. There's no real equivalent of this, and then task it to predict MNIST characters with spikes, and it does work with backprop, and it performs better than a linear network. But an MLP with simple block matrices and nonlinearities, activation function would still be easier to optimize and also outperform this. I think at some point it's also really hard to make a comparison, but this is sort of proof of concept. Then how to translate this to other circuits, I think I already said. We have so much limited access to neurotransmitter, receptor identities, glia cells, modulators, gap junction. We know that a lot of this is critical for circuit functions. So what I think is best to do is, measure cellular ultrastructure, extract morphology, extract connectivity, combine that with function measurements and transcriptomics, and then advance the machine learning methods that chew this data to build powerful models. Quickly going to end on this. So we have a little toolbox also for our paper if you want to check it out, and help contribute whatever. Feel free to raise issues or ask me questions. And yeah, I want to quickly acknowledge everyone who worked on this with me together, Jakob, Srini, and all the people who gave feedbacks for this work, and of course everyone in those two labs that I'm very happy to be affiliated with. Okay, that's my talk.
Yeah, thank you, Jan. That was great. Let's move on to the Q&A. Just a quick reminder to check the workshop guidelines, Alex, in the chat. So that said, any questions from our speakers? Philip?
Yeah, I was wondering, first off, fantastic talk. I was wondering about selection. So like, what if you had chosen, say like image classification, like as a task rather than motion? Like, it seems like somewhat surprising that like all of the visual system can be boiled down into like motion detection. And I'm curious how you selected that particular task and what would happen if different task was optimized for.
Yeah, that's a really good question, and that we get, I think, every time. And I think so. The background is a bit that motion detection in the fly vision system has been, like studied for decades, and so this has been a focus because there are interesting mechanistic models that are very simple reductionist models of how a motion detecting neuron works, and there have been controversies about how a real organism actually would implement it. So these connectomic analysis that we also use the data from, right? They specifically focused also on the motion detecting pathways, and which is the majority of the fly vision system. So that actually, I think, amounts for like 60% of the neurons. And we know, in addition, that behaviorally it's just very important for the fly to escape, to survive. So it was very clear that that if we use one task and prioritize, because it's already hard to train it on one task, right? So training it on multiple tasks may even be harder. then it's motion detection over any other task. But there are also other tasks that the visualization of the fly does, and they are known. For example, color, the color pathways, and then phototaxis and so on. So now regarding what would happen if we just use another task. So we could train it on classification, and it wouldn't work. So I think it might be part of the peer review file of our paper, where we train it anecdotally on depth and autoencoding. And we see that we cannot simply predict neural tuning when we don't have the right high-level task of the circuit. So it's an open question what we do if we don't, and if there is a way to sort of abstract this. And I think that's a very interesting open thread, actually. Yeah.
Randall?
Yeah, thanks for the wonderful talk. I think when I read the paper, you also did a number of studies where you used artificial neural nets to test the impact of sparseness. And I was wondering if you could say something about that.
Okay, sure. So yeah, this was done by one of my co-authors. So we basically tried to make a toy framework for representing this, right? And that there was a recent. So when Srini and Sridharma started this, there was the lottery ticket hypothesis paper. Some of you may know it, which basically shows that one can prune a neural network during training to a certain degree of sparsity to create a neural network with fewer parameters, which can still equivalently well compute a task, which is kind of neat because parameters, like the fewer parameters, the less cost in principle. And that enabled us to make an MLP basically student-teacher framework, where we prune a teacher to a certain degree of sparsity. So it has just 6 layers, and we prune it to various degrees of sparsity. So 10% of neurons are connected, 20, 50, 80, 100. And then we use that connectivity of the teacher and put it into the student network, which is basically just the same architecture, just now that it has precisely the same connectivity. It has the same signs, and it may or may not have some measurements, some noisy measurements of the exact magnitudes of the weights of the teacher, right? And then we can train this student again on the MNIST task, like we trained the teacher from scratch, right? So it has now connectivity constraint, science constraint, magnitudes measured, and learns the task again, which is really parallel to the fly vision system study that we did, right? And then we can ask how well do these students predict the neural activity of the teachers. And what we found, and I might have this figure here. So yeah, this is the setup. It's very equivalent. We take those connectivity measurements. This is feed forward. So it's slightly simplified, but in large parts it's equivalent. And then we compare those predictions with also correlation. And we find that very sparse networks, for those, first of all, we can predict relatively well the single neuron connectivity, and then the denser the networks get, the more connectivity there is, the harder it gets. In particular, if we don't have any measurements of synapse count, so basically some correlate to the synapse strength. If we have measurements of synapse strength or some correlate to it, then even for dense networks, in this case, it seems to work, right? Which is I think a very nice theoretical result. Yeah.
Do we have any other questions from our speakers? If not, we can move on to the general audience Q&A. So, Randall?
Sorry to jump in again. You can also mix mine in with the general questions, actually. But I think there was a point in the presentation where you pointed to the delay for MI4 between null and predicted, and you mentioned, or it seemed like you mentioned, this was quite important. Could you say something more about that?
I wish I have. I'm not sure if I have some of the filters. Okay, so why is this important? So the relatively canonical implementation of the motion detector has two major inputs, and one is excitatory on one side, and one is inhibitory on the other side. And so if now the motion detector here sits in the middle, right, and then the receptors here receive input from above, right, from this plane, and then the edge would first hit the excitatory detector, and that will excite the cell, so the cell depolarizes, and then the edge would hit the inhibitory receptor, and that would inhibit the cell, so nothing happens really, right, because the cell already depolarized. Now, if the edge moves from the other direction, then it would first hit the inhibitory one, and with a delay, which is the important part, that inhibition arrives with a delay. So then the edge has already traveled a little bit further and hit the excitatory one, and the inhibition and the excitation, they arrive at the motion detector at the same time, and so the detector is silent, right? And this makes it discriminate the motion into these two directions.
So whether that works seems to depend then on the speed of motion, doesn't it?
Yeah, that requires a fine tuning of connectivity. So two ingredients, right? Like the actual spatial arrangement of the detectors or of the receptors plays a role, and the membrane time constants, which kind of can represent a delay. And you know, like, I think of it as in the real system, it's there are so many small synapses, right? And these currents and these ions that flow through it, they are very sort of continuous. And so in this way I explain it, it sounds relatively discrete, and it seems to only work for a very specific speed. You know, but I think in the reality, it works for a range of temporal frequencies and spatial frequencies, and then the fly is the vision system is to some degree also able to adapt this range. So with neuromodulation or peptides or something, I'm not an expert in this actually, but yeah, there's a lot going on, and yeah.
Cool. Let's go on to our general audience questions. So someone asks, what would it take to predict the parameters of the system, not through task optimization, but from functional data instead?
Yeah, that's a really good question. So in principle, we can use functional data in the same way, right? So we have from the forward simulation, we get dynamic neural activity, and that is computed in a differentiable way, and we can compare that to functional measurements with some loss function, and then we can backpropagate differences to update the parameters. In reality, we have not so many measurements. So we have 45,000 cells, and then typically we can have, you know, with different recording methods, we have different we have to make different trade-offs. Either we can record calcium across the whole portion of the eye, but then that is that has nonlinear dynamics of the calcium indicator, that has bad temporal resolution, typically, right? Bad spatial resolution. It just captures the whole thing. Or we can stick in an electrode which only measures the single cell, and that has really high temporal resolution but no spatial resolution, and that that might be very precise, but then we also have to match sort of, we always need to know the corresponding input, right? And that is also difficult because we stick in an electrode somewhere, and then we need, in the model, we need to show the correct input at the exact correct location that created this response. So that's tricky. So even if we take all the neural activity measurements that are available in these 26 studies, or even more now, and integrate it in the model with machine learning tools, which is totally a plausible thing to do, right? It is a lot of work to figure out these little nuances of it. And then there is some theoretical work, I think, maybe that goes more into the question of system identification. How many neurons do we need to measure in such a system to constrain the whole circuit? And hopefully, like maybe one of our results hinted at, it's not every single neuron. Yeah. Okay, Conrad said that's what my talk will be about. Great.
I guess we should also follow up with a question asked by Conrad. Most animals whose visual systems I looked at in detail do not perfectly translate, do not have perfectly translation invariant tuning. Basically, the horizon matters. Do flies really have no violation of translation invariance?
Yeah, that's a relevant question if one wants to model the whole eye. So there is so in particular when one gets at the edges of the fly eye, right? Then the orientations they differ. So it's not perfectly planar, right? So yeah, I think to a very good approximation, it's invariant in the central portion, and then if one wants to build the whole eye, then one has to take those invariances into account. Yeah, at the edges.
So before we move on to other questions, just a reminder that you can also raise your hand if you'd like to sort of voice your own question. But until then, I do have a question myself, Jan. So why did you guys use UMAP as opposed to the other dimensionality reduction methods? Because if the goal is to sort of preserve global and local data, why not use something like PacMap?
Yeah, and to be honest, I tried a bunch of things. So I think first I used t-SNE, then I used UMAP, and I found most consistency across UMAP. And then we just settled for it. At some point, I think I also installed PacMap. I think it just came out, and I was excited about the promises that it made. And I tried it, but would have just cost me to rework stuff. And in the end, it's just a method that is a very, very important method in this context. But it hopefully doesn't change the results qualitatively. And that was my impression from some anecdotal trials. Yeah.
Yeah, that's understandable. Thank you. Next up, we have a question from Alexander McLean. They ask, are there any new measurement technologies coming up to allow the kind of ultrastructure measurements needed to capture the missing data?
I'm certainly not the expert in these areas, but I'm excited about expansion microscopy. I think that might go into an answer to this question. And then, yeah, other than that, I cannot think of an answer.
So, Randall, I know you have quite a few. Is there one you'd like to prioritize at the moment?
Let's see here. Yeah. Okay. So going back a bit to early in your slides, you used the average synapse count between types of cells to create your connection matrix. And it's not obvious from that matrix what the variance is, or say the histogram of connectivity. Like, is it really a nice sort of Gaussian sort of curve, and is there a nice average there? And because I'm trying to get an understanding for just how symmetrical, how templated is this fruit fly visual system really?
Yeah, I think that's a very important aspect. In general, also when looking to translate this, I think in the visual system we are in the sweet spot, sort of. I think of it as largely hard-coded in terms of its connectivity across individuals, but even across columns. And I think the reason for biology to hard-code or to make it so crystalline is because its vision is a parallel computation. So we want to be able to detect motion that happens here in the same way we are able to detect it here. And that is basically what we see, that there's this canonical circuit that is basically repeated across the columns, and it is in every location. It is able to do the same computation, and then there is. So I think I want to point you to two studies. One is, I think, the Takamura et al. 2015 synaptic circuits and their variations is the title. And if I remember correctly, they report like 1% variability in this specific circuit. And then Philip Schlegel's paper comparing the flywire data across the hemibrain and the flywire hemispheres. I think that's also very, very nice to see how much variability they find there and how much consistency. Yeah.
Thank you. Yeah, another question I had, which is maybe a bit more kind of stepping outside. By the way, I was very glad that you, in your final slides, that you tried to kind of take some perspective and see where that was all going. One thing that your study relied on fairly extensively as a way of validating and of testing whether or not predictions were good was the fact that there were so many previous studies, 26 or more. What would you do in a situation, let's say you had a subcircuit, some system where there weren't that many studies available? How would you approach the problem then? Like, would you rely on functional recording as the approach? So this is getting a little similar to another question, I think.
Yeah. I think to validate a model like this, we need the functional measurement to some degree, and that can be on different scales, right? Like ideally, we are able to integrate it on a low level, but maybe we can also measure behavior of an animal, and even build some sort of abstraction of that into our model. So as a readout or whatever, or maybe even towards these reinforcement learning versions, and then validate it based on this. So I think it's a multi-scale approach that is necessary and makes it a bit patchworky. Yeah.